Good evening, students. So in this video, we are going to discuss a clinically very important syndrome called Bartter syndrome, right? We are going to discuss all the subtypes of Bartter syndrome, right? And the pathophysiological mechanisms involved in Bartter syndrome. It's a very interesting syndrome. If you understand Bartter syndrome in detail, you are really going to enjoy the various pathophysiological processes involved in Bartter syndrome. So let's try to understand it, right? Now, first of all, let's draw a nephron, right? We will draw a smaller nephron this time so we can have space for drawing all those pathophysiological mechanisms. And right, our descending limb, right? And let's draw a small loop of Henle and our ascending limb, right? Ascending limb and our DCT, right? So where exactly the problem lies in Bartter syndrome, right? The problem in Bartter syndrome lies in your one cell of the thick ascending limb of loop of Henle, right? So we are going to enlarge this one cell and just see, right, what is going wrong inside the cell, right, of thick ascending limb of loop of Henle, right? So first of all, to understand what goes wrong in Bartter syndrome, we need to understand what is going inside these cells of the thick ascending limb of loop of Henle. So we'll take one cell and just enlarge it, right? So let's consider two sides, right? The basolateral side and the luminal side. Luminal side means the side on which you have got urine, and basolateral side means the side on which you have got blood vessels, right? Okay, and we'll start with our main hero. Who is our main hero? Our main hero is our sodium potassium pump, right? It pumps sodium outside and pumps potassium inside, right? So if pumps sodium outside, then the sodium level inside the cell will reduce. The sodium level inside the cell will reduce. And this gradient will be used by which ion? This gradient will be used by our one very important ion, right? One sodium, one potassium, and two chlorine, right? Now this 2 chlorine which is entered inside, right, can escape this cell via two routes, right? One is your potassium chloride symporter, right? It can escape via this route, potassium chloride symporter, right? Other one very important route is your 2 chloride channels over here. Now this is something very important, right? You have got 2 chloride channels over here, right? You mean 2 types of chloride channels here. Of course, there will be many chloride channels. So you got one chloride channel called CICKA, right? Another chloride channel called CICKB, right? Now proper understanding of these chloride channels is very important for understanding Bartter syndrome, right? Now both these chloride channels are further made up of 2 subunits, right? Alpha subunit and beta subunit, right? Even CICKB is made up of alpha subunit and beta subunit, right? Now, why we need to know this? That it will help us in understanding what type 3 Bartter syndrome is, and it will also help us in understanding the difference between the type 4A and type 4B. We'll come to it. But first of all, we need to understand, right? This CICKA alpha, right? It is coded by CLC-NKA gene, right? CLC-NK gene, right? And this CICK beta, the alpha subunit, is coded by CLC-NKB gene, right? So understanding this is important. The alpha subunit is coded by CLC-NK gene, and the alpha subunit of CICKB transporter is coded by CLC-NKB gene. Okay. Now beta subunit is coded by the same gene in both these channels, and name of that gene is BSDN, right? BSDN, both for beta, both for CICKA and both for CICKB, the gene is same, BSDN gene, right? Now we need proper understanding to this for differentiating between type 4A and type 4B, and for understanding type 3, right? And also for understanding symptoms. We'll come to it later, right? First, let's draw whatever is happening inside this cell, right? Now chlorine has got 2 exit gates, right? But potassium has also come inside, right? Now potassium can exit through this potassium chloride symphoter also, right? And potassium can also exit through one very important ion channel called ROMK channel, right? What's the name of this channel? ROMK channel, right? It can exit through ROMK channels, right? So this is the basic of what exactly is happening inside your one of the cells of the thick ascending limb of loop of Henle, right? So now what happens in Bartter syndrome, right? So let's discuss it one by one, right? Let's discuss first of all type 1 Bartter syndrome, right? What we'll discuss? Type 1 Bartter syndrome. Let's write in short form BS. Type 1 Bartter syndrome. It's also called antenatal Bartter syndrome or neonatal Bartter syndrome, right? Because of its early onset, right? Type 1 Bartter syndrome, what it will do, it will affect this ion channel directly, right? Direct effect on this ion channel. You can just see how severe this syndrome is going to be. It is going to directly affect this ion channel. So what will happen if this ion channel gets affected? The sodium won't be reabsorbed. So sodium will be here, right? Now all this sodium will migrate out, go to DCT, go to collecting duct and get excreted out, right? So wherever sodium goes, the water also follows, right? So even the water will be excreted out. So it will lead to polyuria, right? If this thing leads to polyuria, suppose there is a fetus inside a mother's womb, right? And this syndrome happens. Now sodium is going outside, water is going outside. That means that fetus has got polyuria, right? As all of us know that this amniotic fluid inside the womb of the mother, right, is basically urine of the fetus, right? So if the fetus urinates a lot more, so you will have polyhydramnios, lot of fluid inside this amniotic cavity. So this uterus is going to distend because of the excess of amniotic fluid, right? So what it will lead to? Fetal polyuria, okay? Fetal polyuria. What fetal polyuria can lead to? It can lead to polyhydramnios. What polyhydramnios can lead to if the uterus is getting distended, right? What uterus will think that the baby has gone big, right? So the reflex contractions will start, right? And this can lead to premature birth, right? This can lead to premature birth. What else it can lead to, right? If lot of sodium is going out, right? So it will lead to ECF contraction. The ECF volume will reduce because with the lot of sodium, the water is also going out, right? So this ECF volume reduction will lead to activation of RAS. The RAS will get activated. Okay, if RAS gets activated, right, it will increase your angiotensin 2, which will further increase your aldosterone, right? This aldosterone will act on the principal cells, right? And what they will do? They will reabsorb sodium but will end up excreting out potassium, right? So what you will get? You will get hypokalemia. So this kid is going to lose lot of potassium, right? So there will be failure to thrive, lot of polyuria, lot of loss of potassium. So there will be failure to thrive and slow growth, delayed milestones, right? So there will be failure to thrive. And this is also one of the conditions in which aldosterone is high, but still BP will be low. Means aldosterone won't be able to compensate for the overall loss of sodium. It will try to reabsorb some sodium, will end up excreting potassium, but that absorption of sodium won't be able to compensate for this excess loss of sodium, which is caused due to blocking of this 1 sodium 1 potassium 2 chloride, right? So this will lead to failure to thrive. Right? So it's a very severe syndrome, this type 1 Bartter syndrome, right? So what goes wrong in type 2 Bartter syndrome, right? Let's draw type 2 Bartter syndrome over here. Type 2 Bartter syndrome. Now type 2 Bartter syndrome is going to affect your ROMK channels, right? It's going to affect the ROMK channels. Now if these ROMK channels get affected, what exactly can go wrong? This potassium won't come outside, right? And actually potassium is being cycled like this. This potassium will come out like this, and then it will go and participate in this 1 sodium 1 potassium 2 chloride ion channel, right? And the potassium over here, with the potassium which you find over here, is the rate limiting substrate in this ion channel, right? Rate limiting substrate. Why rate limiting substrate? Because it is found in the least quantity, right? So this ion channel needs potassium for its functioning, but if the ROMK gets blocked, this potassium won't come for getting recycled. So there will be, because of the blocking of ROMK, there will be secondary inhibition, right? Secondary inhibition of 1 sodium 1 potassium 2 chloride channel in type 2 Bartter syndrome, right? Secondary inhibition, what we call. Right? So what will happen in type 2? Type 2, the ROMK channels will be blocked, right? So potassium won't come over here. If potassium doesn't come over here, it won't participate in this ion transport. So this will be inhibited secondarily, right? What else can happen in type 2 Bartter syndrome? Actually, this potassium which is being pumped outside creates the positive potential over here, creates a positive potential over here, right? And this positive potential in turn pushes many ions like calcium and magnesium for absorption via paracellular route, right? There are many ions over here like calcium and magnesium, which are actually pushed by this positive ion, right, for its secretion via paracellular route through tight junctions, right? So these two ions are reabsorbed via paracellular route, right? Now suppose this ROMK channel gets blocked, this positive charge will no longer be there. This positive charge will no longer be there since ROMK channel gets blocked. So this magnesium and calcium, right, won't have anything to push them for the paracellular reabsorption. So all this calcium and all this magnesium, right, will be lost. So you will have calciuria and magnesiuria, right? You will have calciuria and magnesiuria. Right? Now, what calciuria can lead to? The calciuria in turn can lead to your osteopenia. The bones will be depleted of calcium. Right? And what else calciuria can lead to? Right? If you are having excess of calcium over here, right? If you are having excess of calcium over here, there is higher probability that this calcium can get deposited in the nephron, right? So what this thing can lead to? This thing can lead to nephrocalcinosis. Right? And what nephrocalcinosis can lead to? Nephrocalcinosis can lead to chronic kidney disease, CKD. And what CKD can lead to? CKD can lead to end stage renal disease, right? So it's a kind of chain reaction, one thing following the another, right? First your ROMK channel got blocked, right? So this positive potential is lost. Now magnesium and calcium has got nothing to push it to the other side for paracellular transport. So magnesium and calcium will get excreted out through urine. You have got calciuria and magnesiuria. Calciuria can lead to osteopenia, right? And nephrocalcinosis. Nephrocalcinosis can further lead to CKD and ESRD, right? The hypomagnesemia, right? The hypomagnesemia of Bartter syndrome is not as severe as hypomagnesemia of Gentleman's syndrome, right? But still the magnesiuria in this case, right, can lead to decreased osteoblastic activity and brittle bones. Right? In case of severe hypomagnesemia, right, it can also reduce the ability of a parathyroid gland to secrete calcium, right? So that can lead to your hypocalcemic tetany, right? It can also lead to a failure of activation of calcitriol, right? Which can also reduce the absorption of calcium from the intestine, right? The severe hypomagnesemia can also lead to torsades de pointis, right, and pseudo gout, many things, right? Now this is for type 2 Bartter syndrome, right? Now let's come to type 3, right? So now this understanding will help us, right, that we have got two chlorine channels, CICKA and CICKB. The CICKA has got two subunits, alpha and beta. Alpha is synthesized by gene CLC-NKA, beta by BSDN, right? CICKB, alpha is synthesized by CLC-NKB and beta BSDN. Now why we need to understand it? Because after understanding this classification, we will be able to understand what goes wrong in type 3, right? Now we'll come to type 3 Bartter syndrome, right? Now what goes wrong in type 3 Bartter syndrome? In type 3 Bartter syndrome, this gene, your CLC-NKB, this gene gets mutated, right? In type 3, CLC-NKB gene gets mutated. Now what will happen if this gene gets mutated? That means the alpha subunit of CICKB chloride channel won't work, right? So this CICKB will become redundant. This won't work, right? Now we need to understand one more concept over here, that in kidney your CICKB predominates compared to CICKA. So this ion channel CICKB predominates compared to CICKA. But in inner ear, right, both these ion channels are present almost in equal quantity, right? So if this ion channel gets defective in kidney, actually this CICKA will try to compensate for it, right, but won't be able to compensate for it completely, right? So it will try to compensate for it but won't be able to compensate for it completely. That means chlorine level, since it is not being excreted out, the chlorine level will increase, right, inside, but won't increase to that extent. So type 3 Bartter syndrome, also called classical Bartter syndrome, the symptoms are mild symptoms. Take a classical Bartter syndrome, mild symptoms. Right? So it will have everything, right? It will have natriuresis, right? Loss of ECF, high aldosterone, hypokalemia, alkalosis, everything. But the symptoms will be milder compared to type 1 and type 2, right? Now what goes wrong in type 4 Bartter syndrome? This classification will help us in understanding what goes wrong in type 4 Bartter syndrome and what actually is type 4A and what actually is type 4B, right? First of all, let's discuss 4A, right? 4A. So in 4A, your BSDN, okay, BSDN will get mutated, right? So this means this beta subunit will be gone, right? And this beta subunit of CICKB will also be gone, right? 4A, okay? So what does this mean? The beta subunit of CICKA is not working and beta subunit of CICKB is also not working. So Bartter syndrome type 4A will inactivate both your chloride channels, right? It will inactivate both your chloride channels. This is something serious, right? If both the chloride channels are gone, think about it. If both the chloride channels are gone, the chlorine inside the cell will increase exponentially, and since this chlorine will have no gradient to enter inside, this ion channel will halt. And since this ion channel will halt, you will have all the things, right? Natriuresis, hypokalemia, alkalosis, polyuria, failure to thrive, delayed milestones, everything, right? So this is something very serious, right? So since the chlorine level has increased and both these ion channels are inactivated. Now think about it. The inner ear has got equal proportion of both these ion channels, right? In type 3, this was getting inactivated, but this was still active. This was getting inactivated, but this was still active, right? So in ear, since both are present in equal quantity, the CICKA can compensate for the action of CICKB, right? So you won't have sensory neural deafness in type 3 Bartter syndrome since the CICKA is still working. What these chloride channels do? They maintain the endolymphatic potential, right? If both the chloride ion channels are gone, this endolymphatic potential won't be there and the transduction because of those hair cell movement won't be there, and this will lead to sensory neural deafness in type 4A. So all these symptoms will be there, plus sensory neural deafness will be there, right? Sensory neural deafness. Okay. And in type 3, because CICK is still working, it is going to compensate in inner ear, there will be no sensory neural deafness in type 3 Bartter syndrome. Right? So what goes wrong in type 4B, right? Type 4A is monogenic, right? Only one mutation is there, right? Type 4A is monogenic, mono, right? But type 4B is bigenic, right? If I write 4B, two genes are required to get mutated to get type 4B Bartter syndrome. Right? Now what are those two genes which are required to get mutated? Both these, CLC-NKA and CLC-NKB, needs to get mutated at the same time for you to get type 4B Bartter syndrome. Right? Now what can go wrong if both these genes are mutated? Right? So this codes for alpha subunit of CICKA, and this codes for alpha subunit of CICKB. So both these channels will be gone. So basically, the ultimate result of both 4A and 4B is same, right? Both these channels will be gone. The 4A was doing it through beta subunit, and this 4B is doing it through alpha subunit. So ultimately in 4B also, both these ion channels will be gone, right? If both these ion channels are gone, of course the chlorine will increase. This gradient won't be there. You will have all the things, natriuresis, polyuria, failure to thrive, and of course, yes, since both CICKA and CICKB are not working, you will also have profound sensory neural deafness, right? So both in type 4A and type 4B, you get sensory neural deafness, right? So up to this point it is clear, right? Now you can see the natriuresis is happening, the hypokalemia is happening, and since lot of potassium is also going in outside, this potassium, body will try to preserve this potassium but will end up secreting H plus ion. So you will have alkalosis also over here, right? Alkalosis. And since lot of chlorine is also going outside, right, you will also have hypochloremia, right? And you will also have hypokalemia, right? So hypokalemic, hypochloremic alkalosis in Bartter syndrome, right? Hypokalemic, hyperchloremic alkalosis happens in Bartter syndrome, right? Now there is also one very important type of Bartter syndrome called type 5, right? Now what happens in type 5 is there is one gene MAGED2, right? Type 5. In type 5, one gene gets mutated called MAGED2, right? Now this gene actually helps in translocation of this 1 sodium, 1 potassium, 2 chlorine transport channels from cytoplasm to the cell membrane. It's a cyclic AMP mediated transport of 1 sodium, 1 potassium, 2 chloride from inside the cell to the cell membrane. So there is no not defect in the gene encoding this channel, but it's a defect in gene encoding which is involved in transport of these channels, right? So genes are encoding for these channels, but the genes which are involved in transport of these channels from inside the cell to the membrane becomes defective. One of them is MAGET2, right? So this MAGET2 actually mediate the cyclic AMP dependent transport of these channels, 1 sodium, 1 potassium, 2 chlorine, from inside the cell up to the cell membrane, right? So this won't be expressed, so all this won't be transported inside. It will also lead to same thing, natriuresis, hypokalemia, right? Alkalosis, hypochloremia, everything will happen. Since the mechanism is different, so we have classified this type as type 5, MAGET2, right? Okay, so it is involved in cyclic AMP dependent transport of these NKCC2, right? Cyclic AMP dependent transport. Of which ion channel? NKCC2. Okay. So let's revise it once, right? So what goes wrong in Bartter syndrome? The main problem of Bartter syndrome lies in our thick ascending limb of loop of Henle, right? What's happening inside the thick ascending limb of loop of Henle? On basolateral side, we have got our hero 1 sodium, 1 potassium, which is pumping sodium outside. So this reduces the sodium inside. This gradient is used by 1 sodium, 1 potassium, 2 chloride ion channel. This chlorine can escape via potassium chloride symporter also, and via these two channels also called CIC-Ka and CIC-Kb. CIC-Ka is made of 2 units alpha and beta. CIC-Kb is also made of 2 units alpha and beta. This alpha subunit is coded by CLC-NKA gene and beta by BSDN gene. CIC-Kb, the alpha subunit is coded by CLC-NKB gene and beta by BSDN gene. In type 1, directly this 1 sodium, 1 potassium, 2 chlorine gets defective, right? So if fetus gets this defect, you will have lot of fetal polyuria, right? So since the amniotic fluid is basically fetal urine, so you will have polyhydramnios. It will stretch your uterus, so you will get premature birth. Since the baby is losing lot of potassium also, so this will lead to failure to thrive and delayed milestones, right? The type 2 Bartter syndrome will affect the ROMK channel, right? So the cycle of potassium will get affected, and this will lead to secondary inhibition of this ion channel. This will also lead to the loss of potassium ion inside this collecting inside the thick ascending segment loop of Henle, loss of positive charge. So there is nothing pushing the magnesium and calcium for reabsorption through paracellular transport. If that push of positive charge is not there, the magnesium and calcium will go and get excreted out. If a lot of calcium get excreted out, you will get osteopenia. If a lot of calcium stays inside this tubule, you can also get nephrocalcinosis, chronic kidney disease and end-stage renal disease. Hypomagnesemia can reduce the osteoblastic activity and can lead to brittle bones, right? Apart from that, the type 3 Bartter syndrome will only affect the CLC-NKB gene. So the alpha subunit of CIC-Kb channel won't be formed, right? So only one chloride channel is affected. So symptoms are mild in this case, right? But since this channel is relatively less compared to CIC-Kb, so it is not able to compensate completely, at least in kidneys. But in inner ear, both these channels are equal in number. So in inner ear, the CIC-Ka compensates for the lack of CIC-Kb functioning, and we don't get sensorineural hearing deafness in type 3 Bartter syndrome, right? In type 4A, right, it is a monogenic mutation of BSDN, but BSDN is present in beta subunit of both CIC-Ka and CIC-Kb. Kb. So both these chloride channels will be gone. Since both these chloride channels are gone, right, the chlorine level inside the cell will increase exponentially. So this will get blocked. Lot of natriuresis, lot of potassium will be excreted out, right, failure to thrive. And since both these channels are gone, the endolymphatic potential will decrease inside your inner ear, right. So there will be the hair cells transduction will be impeded, leading to sensorineural hearing deafness. Same will happen in 4b. In 4b, both the genes, digenic mutation, both CLC NKA and CLC NKB will be mutated at the same time, right. Since both are being mutated at the same time, both these chloride ion channels will be gone. Chlorine will increase exponentially, and also for the same reasons you will have sensorineural deafness inside your inner ear. Apart from all these types, we have got type 5, which is caused due to mutation of the MAGED2 genes. This MAGED2 genes is involved in cyclic AMP dependent of the translocation of the 1 sodium 1 potassium 2 chlorine ion channels from inside the cell to the membrane, right. If this gene gets mutated, right, remember one thing, this is not transcribing for this transport channel, it is just involved in transport. Since the gene which is involved in transport of this ion channel gets mutated, right, this ion channel won't be expressed. So we'll have all the severe symptoms, natriuresis, hypokalemia, right, and alkalosis, etc., right. So hopefully this video has helped you in understanding the very interesting syndrome of Bartter syndrome, right. So wish you all the very best. Hopefully you enjoyed this class. Thank you so much.